Sorry. Oh, and we're recording. Got it. I'm sorry that I'm not with you in person, but if I were, I'd be calling on you. So now I can just leave open-ended questions and hope somebody answers me. Anyway, I'm Willie Buxer. I'm over in the 4444 building, and my lab mostly does functional genomics, which I think you guys chatted about last week or a few weeks ago with Mike White. And so I care, my lab cares a lot about, like when we make changes, when we put variants into the genomes of human cells, what's the impact? What's the functional change? And so, but it turns out that even though I don't have formal training in comparative genomics, I'm genetics and neuroscience, this is something that we use in the lab like every day. And over the past few years, it's something that we've been using more and more. And so comparative genomics, evolutionary genomics is, I think, really important. And I hope that I can share with you guys, ways that people use it. And it's a tool that many of you already use. And I think you'll be able to use more and more. Can everybody hear me okay? Am I being loud enough?
Yeah.
Thank you. So the really quick, you can see the word conservation. So a lot of, that was quick. A lot of comparative genomics has to do with, conservation and trying to figure out what sequences are important and why are they conserved. And, but ultimately, like anything, who cares about it, right? So, here at MGI, there's a lot of clinical workflows going on where, you take some sample, you isolate the DNA, you do base calling, you do variant calling. So now for, a patient that has some terrible disease, you have a list of variants. And you guys probably already know this, but if you just look at the exome variants, how many variants does a single person, any of you, have compared to reference? You've got somewhere in the range of 50 to 100,000 exome variants, just exome variants that you can call from short read sequencing, right? If you look at whole genome sequencing, we're talking about a million or so variants, and if you look at other technologies, there's way more that are much harder to see. So what do we do with that? How do we know which ones are causing the patient's diseases? Well, one approach is to use comparative genomics, use evolutionary genomics to try to, kind of impute, these are the variants that may be more important and these are the variants that may be less important. Certainly that's not the only way to do it, right? I think functional genomics is a great way to do that too, experimentally, but this is an important way. And so people use comparative and evolutionary genomics to look into structure, look at function, look at interactions, how things are regulated. You can actually use it to design proteins, do synthetic biology, and importantly, look at the evolutionary trajectories and relationships between proteins and species and all kinds of stuff. So a lot of uses for it. And again, it's a tool that you guys already use and I think can use more. And so what we're going to talk mostly about today is the concepts. And then on Wednesday, we'll talk about some more of the applications of it. So we're going to review kind of some evolutionary ideas, talk about conservation, a little bit about synteny, and then go into a lot more detail on coupling and coevolution, which is where I think the real meat is. But it's hard to do this without chatting a little bit about evolution. So first question to you guys, who remembers the definition of evolution? It's probably been a while, but somebody shout out. I'm trying to pull up the participants so I can ask a random person. Okay, I'm gonna
change in allele frequencies over time.
Yeah, who is that?
Alyssa.
Hey, thanks, Alyssa. Yeah, so it's a change in allele frequencies over time, and it's not animating. Come on. There we go. Okay. Yeah, change in allele frequency over time. So, and the reason for that may be because of selection, or it may not, right? There's many reasons that you get a change in allele frequency over time, but that's the basis, and hopefully you guys all know that very well. So we're going to do a lot of MSAs, or multi-sequence alignment, in this. And so here's your first one on the left. So here's some nucleotide bases. And what you can see is that maybe this is from 4 different species, let's say. And, positions 1 through 14. and they're nicely aligned. And then you can see, some species or some individuals could be either or have a C, and the others have a G at position 8, right? So there's two different alleles. And obviously this isn't, I'm just showing you a snapshot. Maybe if this was over time, then you could talk about evolution. But so this is the basics. And so, selection, of course, would be that the reason for this is because, there's some kind of a natural selection. And I won't ask you guys the question, but there's kind of there's three versions of selection or evolution that we're going to think about. We're not going to get super fancy here, but the first is negative selection or purifying selection. So certainly something you guys just take a second to write down and remember. Negative selection or purifying selection, this is where you like converge and you get the same sequence, you get the same alleles, you purify that allele. So that allele is somehow beneficial. Changing away from that allele doesn't help you in your environment, doesn't help you for some reason. So negative or purifying selection means you're going to see less change in that region because this allele is really good, is helping you. And then the opposite of that is it's still selection, right? But it's positive selection, that it's actually, you're not purifying to the same single allele, but that this region of your genome, this position, it's changing. So we see a lot of differences happening there. And so there's a lot of, you can think of like activity in that region and trying to find the right answer. So that's kind of a quick way to think about positive selection. And non-selection, right? What's going on in the background or neutral, it's still evolution because there's still changes, but there's mutations, there's random events happening that's causing these alleles to change. And of course, those changes are what selection could act upon if that change is good or bad. And so very quickly, right? Negative is things are more conserved. Positive means there's more changes. And then the background, right? There's no selection, but there's some background mutation rate. That's neutral. And if you like to chat with, Barack Cohen, he loves to talk about null models and, without even having a control, is there some background model that you can test for? And that's what we need in comparative genomics, right? We need a background, we need a null model to say, hey, there's changes going on in the sequence all the time. We want to know if those changes are because of selection, right? Is it because of something functionally important, or is it just background? Is it just neutral? And please stop me anytime. So far, so good, right? So for a coding region, it's pretty straightforward to figure out what is the neutral rate. And what we can do is, formally, you can use something called Ka over Ks, right? Which is the non-synonymous rate of mutations over the synonymous rate of mutations. In other words, right, there's mutations happening all the time. But if those mutations change, from one amino acid to another, right, the non-synonymous rate, that's something that's going to potentially cause, a change that there's selection pressure against. And we measure that with Ka over Ks. And most people can use this very quickly to see, okay, we can measure neutral because neutral is like the synonymous rate. And so if we divide the non-synonymous rate by the synonymous rate, that gives us a better picture of like, oh yeah, here's selection. And if you guys are looking at these numbers, you can actually look at them and say, oh, Ka is lower than Ks. That means that that region is purified or negatively selected. And I'll say that a little slower to make sure you guys are following, right? So if a change that actually impacts the amino acids, right, that's something that's likely if it's under selection and you've purified to just a particular allele, then you expect the non-synonymous rate to be lower than the synonymous rate. Whereas if the region is under positive selection, if you're constantly changing that region, then the non-synonymous rate is going to be even higher than the synonymous rate. And neutral, of course, is when the two are balanced, somewhere in the middle. So getting the neutral rate for a coding region is pretty easy. You look at the synonymous rate and the non-synonymous rate. For non-coding, it's not as trivial because where do we look, right? Where can we assume in our genome, in our non-coding genome, where can we just look to assume like, oh yeah, this isn't, we don't think this is obviously under selection and we think this is. You know, many years ago, of course, people thought, oh yeah, all the non-coding stuff isn't so important, but now we know that tons of the non-coding genome is important. So I'll show you a way in a minute, but it's less trivial. But either way, this is how we're going to get our background, and to be able to measure things like conservation, we need some null, we need some background model, and this is a quick way to think about how we're going to do it. Okay. And just to make sure that I say it explicitly, these conserved regions imply functional regions, right? So if a region is very well conserved, we might assume that it has an important function that's under selection, and therefore, right, if we change it, we'll get mutations, we'll get disease and other bad stuff. And that's what we're often trying to identify. Okay, so let's talk a little bit more about conservation. You guys are used to seeing this all the time in various sequence browsers. Oops, sorry about that. And here you can see a track of conservation. And so just so you know, there is a difference between percent identity and conservation. Percent identity is just very simply asking like how many positions are exactly the same. And conservation is interesting because it has this calculation of like neutral rate, right? We want to compare to some background. So let's look at this in a little more detail. So the first thing is just the multi-sequence alignments in general. So you take your favorite gene of interest and you say, hey, I want to like understand which regions of it are conserved and which regions of it are not conserved. And so, you don't have to really do this manually. You could use a genome browser which builds this in. But let's say you wanted to do it. You wanted to build your own multi-sequence alignment. What species would you choose and what impact does that have on it? And so, like everything else, it's a balancing act. So closely related species, the advantage of those is that you get really good alignment. So if you take two species that are nicely, very closely related to each other, and you look at their alignment, it's going to align really well. But the problem is that there's just, the alleles aren't that different, right, between closely related species. So you need differences to learn something. So a quick example of this is, these are actually all different yeast species that Brock Cohen likes to study. But the pictures of the other animals are here just to give you a feel for like how distantly related, even within yeast, these differences are. So if you take two closely related species, you can see that they align really well. The stars means there's an identity, and there's very few differences, which means it's kind of hard to know that what's going on. We can't learn very much from the closely related species. And then the converse for distantly related species is, well, we could learn a lot, but it gets really hard to align really distantly related species because the sequence is so different that the alignment algorithms just struggle. And so the answer of what you should do is pretty simple, is you should mix together, right? You want to bring in more and more distantly related species, but you have to have species in there that help the multi-sequence alignment software connect everything together. So here's a quick example where we've got three. You still have a pretty good alignment, but now you can see regions that are conserved. You can see this big insertion or deletions, and you can start to learn more about how these things interact when you bring more together. Nothing fantastic yet, just thinking about how these MSAs work and how you can approach them. Okay, so let's get a little bit more formal about this idea of conservation. And we're going to do it in the setting of mouse versus human in just a second. Okay, so here's another multi-sequence alignment, just a two-sequence multi-sequence alignment of some region that you're interested in. Maybe this is some domain of a protein that you study. So I've got some stars there showing the identities, and you can see that there's 11 identities. So, again, like that good? Is that bad? Is that well conserved? Is it poorly conserved? So we need some background to assess that. And so you do that. You say, okay, well, I'm pretty sure that these two background regions are not under selection. I want to compare it to my region of interest. And so here you can see my background regions have 10 identities and 8 identities. And so a very simple score for conservation, and you don't need to write down this equation, but I want you to just get a feel for like how these kind of equations work, right? You just say like, well, my region of interest has 11 identities, subtract that from the average background rate, which in this case is 9. And so this might have a conservation score of +2, right? Meaning that you see 2 more identities than you'd expect to see in the background rate. And so this region of interest may be slightly better conserved. So nothing fancy yet. Now, if you guys were to use software to do this, there's many other corrections and background models that go into these calculations. And I think that's awesome. And by the way, there's like whole courses on evolutionary comparative genomics, which I would highly recommend, but hopefully you get the basic idea here. Okay, so let's use this. So many years ago, people asked like, how much of our genome is under selection? It's a really cool question. And it's nice to know, like, is your gene or some region of your gene under selection or not? And so the way that the researchers approached this was to ask about, they basically took 50 base pair regions and compared them amongst the human genome and the mouse genome, which at the time, those were the 2 genomes that they had available to do this with, since it's been done with many more. And they took about 10 to 20 million regions of these 50 base pair regions, kind of sprinkled throughout the genomes of both human and mouse. And so this is what they saw. So the conservation score is something akin to what I just showed you. And big surprise that, of all those 14 million regions, you get some peak in the this conservation score, and then there's some tails. And what you can also notice is it's skewed a little bit to the left. And they, I think, correctly said that this is likely because there's some region that's under neutral evolution, which is defining the main peak. And then there's a smaller set of genes that are under selection that are more conserved than you'd expect by chance. And so that actually shows you the answer right there, right? And so if you assume that the majority of the human genome is under neutral evolution and is showing you background, then that makes this main peak. And then the side peak says that, about 5% of our genome is like actively under selection compared to mouse with this earlier version of a study. Does that make sense? Questions about this?
I had one question, which I typed in the chat, but I'll just read it out loud now. So I was wondering if positive selection would not be represented because you would expect there to also be a tail with a lower conservation score, but that's represented as neutral.
Yeah,
is it just such a low frequency?
And to everybody who's in here, I'm not paying close attention to the chat, so you can just ask it, but obviously at the end I'll pay back attention. But it's always great if you ask in context. So thank you so much for asking. Yeah, so you're exactly right. So the selected that I drew here is purifying selection, is negative selection, and that's what they're seeing. It's hard to see, which would be on the left-hand side. So that tail over there, I drew the red into that tail, but I agree, there would be some region over there that's likely under positive selection. And it's kind of hidden in the tail to the left, is my guess. I actually think that there's been many more newer versions of this kind of study with more genomes, and they more directly ask about positive and negative selection. But you're absolutely right that it is there, and you would see it, somewhere else in this graph. Honestly, this way of calculating the conservation score, it's a little bit harder to get at positive selection. But if you had many different individuals, instead of just one human genome and one mouse genome, you would have a better time calculating. But yeah, great point. Okay. And this is a 2002 article, a little dated. Okay, so we talked a little bit about conservation. So let's try to use it. And again, many ways you might want to use this. Most comparative evolutionary biology, it's going to be hypothesis generating, right? So you're not going to solve the question. You're going to get really good hypotheses that you can then go and test in other ways. And so going back to my initial kind of proposal of like, here we have this patient that has, let's say, a neurodegenerative disease, and they've got 50,000 exome variants. Can we use conservation to ask which of these variants may be the cause of their disease, knowing, without any other knowledge? And by the way, that approach I put up in the top left corner here, the phrase lookup table, is one of the big efforts that WashU and other schools are undertaking, right? Can we have a lookup table for every possible variant that a person may have and know, like, is that variant benign? Or can you tolerate it, or is it pathogenic? Does it cause a disease? And so, we want to do that functionally, right? But can we also use comparative genomics to help us figure out which ones are which? At least generate hypotheses about that. And so here's a paper that tries to do that. And can we use these conservation scores to try to figure out what variants might be pathogenic and which might be benign? And you guys might know there's a lot of scores, like CAD scores, that try to make those kind of predictions. So here's a local example that really uses evolutionary biology. And so they use two factors in their model. One is called shared taxa, and the other is called local identity. And so here you can see a table that has Homo sapiens and rats and mice and cows and pigs and goldfish. And it's just listing for each of those species how many taxa are shared. And so when you make these comparisons, you can list, like, basically going up and down the tree, the phylogenetic tree, how many shared taxa are there between any two. And then local identity is just identity. So here's how this works. So on the top, you've got your reference sequence that happens to be in amino acids. And then on the lower line, you have your query sequence. And you can see that there's a variant here, right? So you have a missense variant, it goes from S to A. Okay, so now you say, okay, now I'm going to compare sequences so with really high shared taxa, right? So I'm going to look at, let's say, another human or a human reference sequence. And you see, okay, there's, of course, this allele, and then there's this other allele. So the local identity is 7, right? There's basically 7 blue boxes here, and the shared taxa is 31. Okay, and then you say, okay, I'm going to look at another animal. Let's say I'm going to look at a chimp. Okay, here's a chimp sequence. There's 8 identities, so Li is 8, and the shared taxa is 29. Right, so you do this for all the different species that you have in your multi-sequence alignment, and you get both the local identity, which of course, as you get further and further away of your query, which in this case was human, right, there's the shared taxa goes down, and then also the local identity is going to drop because you have less of the sequence is exactly the same. So they built a model just using those two numbers, and they called it the variant shared taxa score, and it's not bad. So just using those two things, they were able to take a set of variants where we know that they're pathogenic and we know they're benign from other data sets. And here you can see how well they classified it. So high VST scores were likely to be benign, and low VST scores were likely to be deleterious. And so you can see that the grays and the blues, they're different, but it's not perfect, right? So it's not enough just to know the shared taxa and the local identity. You can't fully figure out whether something's pathogenic or benign, but there's information there, right? And so this model based on these two very simple concepts already yields a lot of interesting information. And many of the scores that you guys know about, like CAD scores, for example, use various kinds of conservation information and evolutionary information, to try to make their predictions. And so but just keep it in mind that this is a great place to start, but certainly is not the final answer. Any questions on this, like how you can use these conservation and evolutionary information in this setting?
Can I ask a little bit of a random question? I was curious if you thought maybe like when we're looking at coding regions, if one day we'll hit a point where you can just look at these coding regions and the differences between them and really be able to predict how the protein was like changed over time. And just have like more definition to it.
I think so. And it may actually be something we hit on in a moment as I go a little bit deeper. So ask me again if I don't answer that question exactly. Okay, and so I think this is the last bit I have about conservation. So like we said, you guys can check this very easily by going on to any kind of a sequence viewer. There's usually a track that pulls up an alignment, a multi-sequence alignment. You guys can make these manually. The advantage of making it manually is you can pull any species or any individuals you want to try to look at these comparisons. And so this is another paper that came out after the rat genome was sequenced, looking at the human, the mouse, and the rat. And they found that we'll just do the bottom line here, about 5,000 sequences that had perfect like identity, right? We're 100% the same, that were at least 100 base pairs long. And so these are like ultra conserved sequences. And as you can see, there's also longer sequences that are ultra conserved amongst human, mouse, and rat. But it's not, it's not millions, right? There's 5,000 regions that have that really good conservation. And here's what it might look like in a genome viewer, a genome browser. This is the Maltese alignment that has, human, chimp, mouse, blah, blah, blah. And you can see this conservation score is calculated by that MSA. And you can see regions that are really highly conserved like this one here. And, there's not a big surprise that in a coding region like this exon that there's pretty high conservation. But what about here? What about this, which is the region I zoomed up front? This is a non-coding region that's really, really well conserved. And this is how, one of the ways that we identify regulatory regions, which you guys know lots about. So promoters and enhancers and things like that tend to be really, really well conserved and are included in these 5,000 or so sequences that have like perfect alignment, because this is where other proteins like transcription factors come in and bind. So that sequence has to be really well conserved to get that protein to come in and bind. And so again, the point of all this is you can use it. You have your gene of interest. Maybe you have variants that nobody knows about. Like, what rules do they break? What rules do they, continue to adhere to? So, but big surprise, regulatory regions are very well conserved. Okay, synteny we'll do fairly quickly. Synteny is just the preservation of order. So conservation has to do with, we usually mean the conservation of sequence or sequence identity. Synteny has to do with the preservation of order. And so it might be the ordering of domains within a protein, it might be the ordering of genes along the chromosome, but any kind of preservation of order is a measurement of synteny. And so here's what this looks like. This is a synteny built around this gene called SARM1, which is a gene important in neuroscience. And you can see that on the genome in, I guess, cow is on the top track here. So on the genome in cow, you can see that it's, vitronectin, SARM, this solute transport, FOXN1, they go in this order. And then you can see amongst other mammals, which is like the top half of this, those genes tend to stay in the same order, right? So like the colored lines are pretty nicely conserved as you go down. So the actual position of these, right? They might be millions base pairs further apart, but it just says that there's not an intervening gene in between them, or if they are, right? Like here between SLC and FOX, they're usually right next to each other, but here happens to be two intervening genes. but then still Fox isn't much further away. It didn't like disappear and go to another spot. Everything stays pretty close together. So that would be an example of a syntenic region or a syntenic unit or a syntenic cluster. And the assumption is that there's some reason that it's not ideal to move these apart, right? If you move these away from each other, it might have negative consequences, and that's why they've stayed together in this format. We don't know why, but again, it implies that something important about them being together. You can also see now, if you look below the line and skip to, there's several avian species on the bottom here. You can see that they are also conserved. The core of this, right, from the blue all the way to the pink, is pretty well conserved. Or sorry, that they have high synteny. But then starting to the left of this blue line, mammals have a different syntenic unit, right? That's this ugly green color going left. Birds have a different set of genes right here. And, not hard to imagine how this happens, recombination of the chromosomes, but those two syntenic units are different. And so that's it, preservation of order. A paper that recently went over this found that in mammals, something like 90%, 87.5% to be precise, 90% of our genes are in one syntenic unit across all the mammals that they looked at, right? So they had a set of mammals like I just showed you, and they found, pick your gene of interest. It's probably one of these, right? Because about 90% are. And it means that amongst all mammals that they looked at, it always has the same neighbors. So has very high synteny. But then about 10 or 15% of our genes, again, amongst mammals, have two syntenic clusters. So what that means is that in some mammals, they have neighbors X and Y. But in other mammals, they have new neighbors. They have another set of neighbors. But they're just in two syntenic clusters. And so here's some examples of those. So BRCA2, TRAP2, SNPJ. And what they're showing is that the cluster on the left, like in the majority of mammals, they have these neighbors, but in other mammals, they have this other set of neighbors. And so they've switched. They've gone to a different lunch line, basically, in this other set. And what this PNAS paper goes on to say is that those genes, the ones that are in their own, the two or more syntenic clusters, they hypothesize, they think, they show some evidence for this, and I think it's probably true, that these are genes that are really important for things like development of brain size and some of the traits that are, may change, are likely to be under selection, probably positive selection. And so these genes kind of switching around may have been important for the positive selection that has changed mammals over time. So cool things that you can kind of figure out from comparative biology. Another side note is that this is true for mammals, that like 90% of genes are have the same neighbors when you look across mammals, but it does not hold true for other clades. So for example, in plants, this would be the far exception. Even within like groups of closely related plants, you have much more syntenic divergence, I guess you would say. So anyway, just so you know, the 90% is a rule in mammals, but not a rule when you leave mammals. Any questions about synteny, preservation of order?
So I have a question about that. I was wondering if, based on synteny, if you can conclude that the order of genes is necessarily important, or rather that there's just generally a selection against large variations, like in chromosomal translocations or inversions.
Yeah, so you'd have to start with getting a feel for like, what's the null model, right? Like we know the reasons why synteny might be disrupted, right? You could have a breakpoint that happens there. You could have recombination that happens there. And there's rules around that, right? There's certain sequences that are more likely to have a recombination event or because of chromatin, and so you'd use that to generate some kind of a null model of like, okay, these regions are more likely to have a syntenic disruption. These regions are more likely to maintain synteny. And then from that, you'd say, okay, now here's an example does that example like fit with our expected null model, or does it deviate from it? With any of this, I wouldn't go so far as to say, certainly I think the way you phrased it is a good hypothesis. Like, we hypothesize that this order, like maintaining this order is important, and breaking it might be harmful. But I'd want to do the experiment before I'd go beyond the hypothesis, if you will.
Okay, thank you.
Yeah. So, but again, more toolboxes, more tools in your toolbox. So check out for your gene of interest. Is it in one of the syntenic clusters that, the 2 or more, or is it kind of like most other genes? Okay, so it's a good second. I'm going to take like a 1-second break to drink. You guys should as well, because we're going to spend the rest of the time on coupling and ideas around coupling and coevolution. I guess it's also a good time for me to check the chat. Okay. And if you're sitting, it's a good time to stand. If you're standing, it's a good time to sit. Change it up. Where is my, okay. Okay, so we've spent the majority of the time talking about conservation, and the conservation is often focused not just on a region, right, but down to a single amino acid or a single base pair, say like, hey, there's an identity here or not. But now we want to zoom out a little bit, and the reason that we want to do this, I knew I put this here for a reason. Okay, in the top right, before we get into coupling, I have a few words that you guys know: Mendelian, monogenic, and polygenic. And it kind of is just trying to approach the logic of coupling in general. So, most diseases are not Mendelian or monogenic, right? In other words, a single variant or a single gene, it's pretty rare that a single variant, a single gene can give you a disease. There are many rare diseases that are that are monogenic. But most common diseases in our population are polygenic, right? They're caused by multiple problems. And that's for a good reason, right? I mean, we have all these genes and they make all these proteins. And our system is really good about dealing with things getting broken, right? So we can tolerate some things getting broken, and it's not enough to cause most diseases. There's a few key things that you can't mess with, but the majority of our genome, while important, just putting a single break in it is not enough to take us down, right? We, evolution has built in enough redundancy and enough interactions to deal with that. So Mendelian and monogenic diseases are much more rare. And similarly, I think we got to approach evolutionary biology and variants in the same way, right? So you guys probably have heard the word epistasis, right? That multiple variants can interact with each other. And we sometimes talk about that as an afterthought, but really that's the rule. All of our genes interact with almost all of our other genes, right? Every day you find a paper that finds a new interaction. So our bodies are defined by interaction. So we should really approach, looking at variants and looking at alleles the same way, not just on their own, but how they interact with others. So anyway, that's my start to get you thinking about, we should never look at something in isolation. Big surprise. And this idea of coupling. Okay, so conservation looks at one region or one base and say, are you conserved? And the hypothesis is that if you're conserved, you're probably under purifying selection, meaning that if we change this position, it's probably going to break something. And so that hypothesis is a good one and has been borne out many times, right? So super highly conserved sequences tend to be bad to mutate. If you get mutations there, there is a good chance they're going to be deleterious. But the converse is not true. So just because a region is not clearly under really high conservation does not mean that it's not insanely critical. In fact, usually it is, but it's harder to measure. And coupling is one of the techniques you can use to measure it. So you guys have seen these kind of logo plots before where the size of the letter indicates, whether it's conserved or not. So you can see at position 10, there's a really big L, right? So that L is saying that like across whatever multi-sequence alignment they did, they always had an L at that position. And so it's really highly conserved. And same at position 2, there's an F. So clearly, if you mutate the L at position 10, you might have bad consequences. And in this case, you do. But what about position 9 or position 8, let's say? So you can see at position 8 that there's a lot of letters there, and they don't add up very high because it's hard for us to understand what's happening at that position. So if I get a mutation there, is that going to be bad or not? And from this plot, we don't know. But that's what coupling is going to help us evaluate. So let's do our own kind of multi-sequence alignment. So let's say this, as always, could be either different species or different individuals of the same species. Let's say it's different species. So we'll do coevolution. And what you can see is that these are all aligned. And so at some positions, you have very high conservation, indicating by these blue bars. In fact, you can see that the really high blue bars are invariant, right? So every sequence we have in the alignment is a C, just like the L at position 10 or the F at position 2, right? So those are the invariant positions. They're perfectly conserved at those positions. And in other positions, there's a lot of change. And then there's some that have like a little bit of conservation. So it turns out that with coupling, we actually don't know anything about the invariant positions. Like they're kind of boring. We can't learn anything about like the cool part of coupling. We kind of have to ignore those. And so sure, we know they're conserved, but we can't use them for coupling. But it's all these other positions that have changes that are the interesting part. And so if I showed you guys these two positions, so the ones that have C and A and the ones that have T and G, do you guys notice anything interesting about this? Anybody want to point out what could be going on here? What's unique about these two that I highlighted?
It looks like the C's are always paired with T's and the A's with G's at these positions.
Exactly, right. And so she said that the C's are kind of paired with the T's and the A's are paired with the G's. So even though the letters themselves don't matter, but the point is that when these animals, basically when there was a change in the allele from C to A, there was a subsequent change from T to G, right? So the evolutionary history is kind of apparent here. And so it's like you needed something about changing from a C to an A maybe required a change from T to a G. And that implies that these two, so certainly they co-evolved, right? There was, if you were to measure correlation or coevolution, these two considered together would have a high score, right? They're highly, in some ways they're correlated. And the implication there is that they're somehow interacting with you. Maybe they're physically connected. Maybe the amino acid they produce, like it has to be a plus and a minus, or it has to be two neutral. But whatever it is, this implies that they, there's a chance that they may be interacting because of this coevolution. And this turns out to be really cool because now we can do all the pairwise comparisons. And this is what I've done here for a protein that I'm interested in called SARM1. Every line here, so if you were to read across the bottom, these would be the first, let's say, 300 amino acids of this protein, just in order. So amino acid 1 through 300. And then it's the same amino acid 1 through 300 going up. And the color of, within the matrix is just the coevolution score. And so to keep it simple, it's just the correlation, just like you pointed out here. If there was, and this is I think it's 100 species alignment. So we're looking at like 100 columns and we're saying, is there a correlation between the changes that happen between, position, let's say position 1 and position 150, or position 1 and position 50, right? and you build up this matrix. And it's really cool, right? So if I knew nothing about this protein, I would say, hey, it looks like there's a domain right here. It looks like there might be another domain right here. And then maybe there's another domain at the end. So it kind of looks like there's 3 domains. I might also say, hey, it looks like domain 1 and domain 2 are interacting, right? Because see, there's like this darker blue color here. And then there's this region that's like really hard to tell what's going on. And then here, domain 3, and you can see like within the domain, like they interact with themselves probably because they're all like folded together into a nice domain. But what's also super cool is that you say like, hey, look, it looks like domain 3 interacts with domain 2 at this site, right? Do you see this little blue region off the axis here? So maybe this is where these 2 domains actually sit together and connect together. And again, we're generating that hypothesis. That's not exactly true, but it actually is close to the physical structural reality of this protein. And so you guys can do this, right? So I made a multi-sequence alignment and I didn't use any fancy math. I just said, are 2 positions correlated with each other? And if they were, I just put that on a scale and plotted it out. And it literally lights up the way that this protein, we already know this protein is structured. So again, there's so much utility here for learning and pulling out interesting parts of this. So anyway, I think coupling and coevolution is really where a lot of new stuff is going, and we're going to go into some of that new stuff. But do you guys get the concept? And does this plot kind of make sense? Okay, so let's go a little deeper. So, just looking at correlation of changes isn't exactly the best way to do it, right? Because we need some kind of a null model. We need a better way to calculate this. And and of course, the big picture here is that just because something is not well conserved doesn't mean it's not important. And in fact, we get more data from when it's not super well conserved because we can do this. Okay, the formal way to do this is called SCA, or statistical coupling analysis. And one of the groups that have worked on this that I think is doing a great job is out of the Ranganathan lab. And so this is a paper from 2016. But it's basically exactly like what I just told you. And so on the left here is just like the one that I showed you, but within a kinase domain. So rather than looking at the whole protein, they're just looking at a kinase domain. And so here's the positions within the kinase domain. And you can see that some positions have, more or less coevolution signal. And here, instead of just looking at correlation, they're using a more complex way to calculate it that's more accurate. And so you can see within this, domain that there's regions. But we don't really care about the positions in order. In fact, we know that the 3D structure is way different than the, like the order of the primary amino acids. And so you want to decompose this matrix to like figure out what information is inside of it. And here's one really easy way to do that. Just do hierarchical clustering on this matrix. And you can see that within the domain, there's a few regions that really highly interact with each other. And then, less and less interaction as you get further away. And this is just a way to visualize it, but they use a spectral way to deconvolve this matrix. And what you find, so you decompose the covariation matrix, and what you find is what they term sectors. And so sectors, when you're doing statistical coupling analysis, right? So you're asking, how do different amino acids co-evolve? What is their covariation? And then you build up that covariation. You don't just care about pairwise, right? You want to know, like, what are the units of covariation? And those units are called sectors. And usually a domain will have a few, 2, 3, 4 sectors that all co-evolve independently, but within a sector, they co-evolve together. And so here's what this looks like in their paper where they took a kinase domain. So they defined 3 sectors. And again, a sector is not a domain. It's not primary sequence. It's not secondary sequence. It's something brand new that this kind of coupling analysis reveals. So here's what the red sector looks like. Here's what the green sector looks like. Here's what the blue sector looks like. So they went through and they found, people have done a lot of mutagenesis on these kinase domains. And they said, you know what? Pretty much every time we see in the literature mutations in the red sector, that has to do with catalysis. And whenever we see mutations in the green sector, that has to do with substrate specificity. And whenever we see changes in the blue sector, that tends to have to do with kinase regulation. In fact, it turns out to be like insanely, like those statements, like red is catalysis, green is substrate specificity. It turns out that this is like insanely accurate. And they went and they did further mutagenesis to show this. And so these sectors are really pulling out functional units that co-evolve. And if you mess with one of them and you don't have a cognate mutation in another one, you're going to break the protein. But if you have multiple changes within the sector, you can compensate for that and still function just fine. And looking across the evolutionary history of this protein pulls out those functions. And you can, obviously from SCA analysis, you don't know what they do, but you know that there's these co-evolving sectors. So questions before I show you a little more?
I have a question. So when looking at the plot, I think I'm not clear how to define a domain or a subdomain and a sector. Like, what is like the smallest unit that you consider? Is there anything like any general rule or something?
Sure. So if you guys can actually download this. It's both in Python and in MATLAB. And in there are places to set a threshold, which is how you ultimately define it. Remember that you're measuring the covariation of pairs of amino acids. And then, so you're measuring every possible pair in either the whole protein or the whole gene, if you want, or within a domain. So in this example, I'm only showing you, or they only did within a domain. So everything they're doing here is subdomain. And then, like you said, they had a cutoff, or like I think you're realizing is you need some kind of cutoff to say, like, when does a sector start and end? And there's a little bit of math around that. And if you actually run the MATLAB or the Python program, there's a spot where here's what they recommend, but then you can adjust a little bit. And so the sector is trying to bring together all of the co-evolving pairs and mapping them onto the positional space to come up with these sectors. And so there's a bit of math and a bit of thresholding that goes on to actually define a sector. But to be clear, a sector is smaller than a domain, and it's the set of amino acids that are co-evolving up to some threshold. And usually you're trying to find independent sectors that don't overlap. In other words, like you might have 2 regions that co-evolve. Of course, within each region, they co-evolve really well, but then they're connected. And so you could have subsectors. This is a fairly new field, so I think those rules are still evolving. But if you're on the software, you will see where you can set the threshold. Any other questions, or I can try to go deeper into that one?
Yeah, actually, I do have a quick question.
Go ahead.
So the clustering of that map, is that what allows you to identify these individual sectors?
Yes. So one way to do it would be, this is an example with hierarchical clustering, and you'd say, oh yeah, here's one sector, here's a second sector, here's a third sector. What they actually do in the paper is a little more complex. So they use, now I'm forgetting if they use a spectral decomposition approach. I think it's SVD to better define these, but that's the idea is you cluster together the regions of coevolution, and that's what builds the sectors. So it's akin to what you see here, but a little bit, it's not exactly that.
Thanks.
So sorry, going back to figure
Sure.
D. So you see kind of this line going off of the top cluster, like a horizontal. Would
Yep.
that imply that the first and third sectors are also interacting somehow and co-evolving?
Yes, exactly. In fact, whatever, and again, this is not their exact sector definitions, but you're exactly right that it looks like this sector and this sector, the last one, have a lot of interaction between each other. And there's a little bit of interaction between the second sector. But yeah, you're exactly right. So you would, and that's where the thresholding comes in. Like, where do you say like, oh, I just want to combine all these together into one big sector versus you want to pull them apart into 3 separate sectors.
Okay, thank you.
Yep. Okay, so this was the part that blew me away when I first saw this paper. So there's a lot of dots here, but here's the wild type. So wild type has a certain TM. So this is like the stability of the protein. And then it has a certain amount of enzyme activity. And so where 1 is good enzyme activity. Okay, and so now we're going to mutate it. And so the red, this is a particular point mutant that's already known. And you can see that it only moves on the x-axis, meaning it only affects the catalytic ability and it has no effect on the protein stability. Okay, does that make sense? So it shifts to the right. On the other hand, here's a mutant in blue that affects C157A. It doesn't affect catalytic ability at all. Like catalytic ability is fine, but the protein stability is compromised. So 2 different mutants that affect in 2 different directions. And here's a third for fun that, you know, is kind of affects both a little bit. So here's the really cool thing. Now we're going to look at the white circles. So the white circles are examples where you now add together the 2 mutants. So you're adding together 216A and 210. And all this was kind of predicted ahead of time by Scott. And so you'd predict this to be additive, right? Where you have a little bit of an effect from the stability and a little bit of effect of the catalytic ability. So you'd expect this to be here. And when you actually make those paired mutants, this is what you get. And so they went through and actually said, based on their sector predictions, they could start to predict where the mutants would fall in this diagram, first predict it, and then they would go and make them. And then they were usually like insanely accurate to where these would fall. So once you kind of understand what one of these sectors do, you can understand, you can at least hypothesize what the other mutations and other members of the, you know, just so long as they're independent, right? So like one mutation within anybody in the sector is going to, where it's going to fall in this diagram. And then if you make a second mutation in a different sector, it like is perfectly additive. So this is just really cool. So the ability to just from a multi-sequence alignment of your gene of interest, you can actually start to do really cool things for predicting how different mutations are going to affect the function of the protein. Does that make sense? Okay. And so what can you do with this? And so this is a paper that came out a year or 2 later where they're trying to put together ways to, and this, by the way, is going to be the theme as we carry on going into like structural space. But can you use this to design proteins? Can you use this to help with structural predictions? And that's what this paper does. So it tries to like figure out how you can like stabilize different mutations. And people were using energy or physics-based methods to do this previously, right? Say like, oh, we know stuff about how proteins fold. But when they used evolutionary-based methods, it worked much better. So understanding these coevolutionary relationships really helps understand how like proteins fold. And we're going to see that again. So with a much newer paper. Where I think this is the coolest is building this coevolutionary information into functional genomics. And so this is Kim Reynolds, and she gave a talk last year, a keynote talk, and it was all focused on allostery, but she used this exact stuff, right? Here you can see that MSAs give you the covariation matrix, give you these sectors of, you know, what she says, collectively evolving amino acids. And the sectors are sparse, physically contiguous, and connect active sites to distal surface sites. So this is really cool. And so, right, here's the active site, but then through the sector, you can see where that connects to sites on the surface. The idea being now if other proteins come in or you make mutations on those surface sites, it changes allosteric interactions and how you can regulate proteins. And so her proposal in the beginning of this talk was that, messing with these surface sites that are connected to a sector, connected through a sector to an active site are the perfect place to, engineer allostery. I'm using lots of new terms, so I should probably step back. Do you guys understand how, like, if you find a sector that contains a known active site, right? Like, let's take a kinase domain. We know what the active site is, right? People have already figured that out. It's these amino acids. So now you do your SCA analysis and you say, okay, the active site is part of sector 3, let's say. Okay, sector 3, it has the active site, but then it also has this other part of the protein, it has these other amino acids. And so her hypothesis was that if you follow the sector connected to the active site out to the edge of the domain or the protein, right, the surface, that's the prime site to regulate the active site. That's where other proteins are going to come in, or you'll have other domains within the same protein. That's where you should look for regulation. And that's exactly what she did. So she did kind of an MPRA style study, like a functional genomics study in her model system with this DHFR and LOVE2 fusion, and did every saturation mutagenesis, right? So like deep mutational scanning, and then measured the growth rate. In her system, she could turn this into a growth assay. And what she found is that, yeah, when they made mutations in these predicted regions, these regions that involve this sector, they were able to like enhance allostery, they were able to disrupt allostery, and basically these were either depleted in the sector or inserted near the sector, but she was able to figure out this protein and how it interacts with another protein with these sectors. And so again, this is a really cool, really new tool. And if you guys want to watch her talk, you can see a lot more details about it. Sorry about the zooming there. But to wrap up this bit of it, right? You take your protein, you find the sectors, and you use those sectors in combination with things like saturation mutagenesis, deep mutational scanning, or known variants and what they do to the function. And you can really build up what is the function of other variants based on what sectors they belong to and based on their coevolution with known things. So you can really build up the function of your entire protein, your entire gene, and start to predict out with much, much more accuracy than just using conservation. What is the exact effect going to be when this mutation pops up in a patient? So questions on any of that? We have one more thing to chat about briefly.
I have one question. Has anyone ever tried to do a similar type of analysis with non-coding regions that they think might be interacting for gene regulation?
That's an awesome question. So I've done a little bit of that. And so the idea, right, is that it's kind of passé to do it on the protein, like, the coding regions, because we know a lot about the proteins already. But so there are some papers trying to do SCA on non-coding regions. I think that's so cool. And I've tried to do it, and I've learned a little bit about proteins that I've been interested in, of like possibly regulate. But what I haven't done is actually like gone and made the mutations and see if my predictions are right. I haven't seen a great paper, but it's something that I should look for. But yeah, I think people are doing it, and I think it's a fantastic idea.
Okay, thank you.
Okay. So since it's really closely related to this, I want to give you one other example, which is AlphaFold. So you guys have all heard about AlphaFold. Just just so you know, and you don't get confused, there's actually the old AlphaFold, AlphaFold 1, which came out, I think, in 2018. And then the new AlphaFold, AlphaFold 2, which won the CASP competition in 2020. And so, and of course, AlphaFold 2 even has more versions of it. But AlphaFold came out of DeepMind, which is a Google Alphabet-backed machine learning company. And as you've all heard, it's awesome. So we're getting close to the end of the hour, but so I'll just tell you, but I wanted you all to think about what you need to be able to do structural predictions. And you need a few things, but the most important of which turns out to be what we're talking about today. So one of the reasons that AlphaFold is so good is because it uses everything that we've just chatted about. So it uses evolutionary history of proteins, pairwise evolutionary correlations, and it uses those things to figure out homology to solve structures. I actually took these phrases straight from, I forget now if it was the abstract or the intro of the AlphaFold 2 paper, but so things that genome sequencing has enabled, so all of the data that we generate in genome sequencing lets us do this, right? Lets us generate really complex, really long evolutionary histories. So we can build up these really complex covariation matrices, right? As in SCA analysis. You can't do that if you only have like 4 sequences, right? You can't get enough coevolutionary information to find out anything interesting. You need to have what they like to call deep coevolutionary history, right? Really long MSAs to really build up these relationships. And of course, you need an engine that understands physics of like how the amino acids pack together and what kind of energy constraints. And so certainly, like I just showed you, there's people that have tried to do protein folding just with genomic sequencing data very recently. It works pretty good. And for many, many, many years, people have been using physics and energy-based models to do structuring, which works okay. But when you combine them together, you get fantastic results. And that's what made AlphaFold so strong. The third thing, and I don't want to diminish this, is that they were able to basically invent new machine learning around some of these problems, DeepMind, right? And this is a big part of it as well. And if you're curious to think more about that, one of the big pieces of this was the attentional network, which this is important in machine vision, right? Where you have a scene, but you only care about the car, the person, or the stoplight. So you have to, focus your attention or the attention of the model on some part of a larger thing. So they were able to build some really cool transformers and attentional things. And then one of the hardest things for solving structures is like, what are you going to use for your loss function? Like, what is right and what is wrong? Like for mutants, when we talk about variants, it's pretty easy. Like we have a training set where here's the known pathogenics and here's the known benigns. But like for protein folding, how do you know when you make like a slight rotational change in one of the bonds between amino acids? Like, how do you calculate whether that was a good thing or a bad thing, which you need in machine learning to train the model? And so the way they built their loss functions and the way they built intermediate loss functions so they can make small changes, this was another big thing. But so anyway, so people, make a fuss about AlphaFold, you can say like, hey, what enabled that was like evolutionary genomics and being able to link, all of the things that I just talked to you about to understood structures and to build this together in a really cool deep learning model. And so here's a picture of how the model actually works, which we don't have time to go in detail. I'll play this video in the background of, this is like as the deep learning is going, it's calculating these rotations and trying to refine. And you can see pretty quickly, it gets to a decent structure that then gets refined more and more to get to like insane atomic accuracy. And of course, there's lots of papers about this. And since AlphaFold, there's been, AlphaFold has problems as well. You guys might know about RosettaFold, which is from the Baker Lab. RosettaFold is kind of even more impressive because like the Baker Lab, although a huge important lab, certainly doesn't have the resources that DeepMind and Google has, and they're able to get to really phenomenal things. And I think this link that I have right above Rosetta Fold is to another Baker Lab paper where they try to calculate every possible protein-protein interaction in yeast. And again, using these coevolutionary principles. So that's where we'll end for today. I'll stick around if you guys have questions. So we talked about evolution, think about conservation, synteny, the ordering of things, and then a lot more detail about coupling and how we can use coevolution to really exquisitely rebuild the domains and the structure of proteins, and even use that to solve like one of the biggest problems of all time, how do proteins fold? So thank you guys, and please stick around and ask questions as you see fit.
Thank you.
Hey, thanks for the questions. I also listed my email on here, so feel free to email me any questions that you have.
I actually have one more question.
Go ahead.
So going back to the Ka/Ks ratio that we briefly talked about, it's been a while since I took Bio 101, but I guess I'd imagine that, you know, at any given point, you have probably a bit of both positive and negative selection going on at the same time, maybe at like one locus that you're interested in. Is that possible? And does it make it difficult to use the Ka/Ks ratio to sort of deconvolve what's really going on?
Yeah, I have a figure about this in one of my papers. So if you come up with a window size, right? So you're saying like, I'm going to consider, you know, let's say 9, you know, a region that has 9 amino acids, let's say. And you can kind of look through and slide that window through and ask, what is the Ka/Ks? You can like make a profile of Ka/Ks. You will actually find that like within, let's say, a domain, there's regions as you window through that have, you know, neutral Ka/Ks, and then like it'll suddenly spike down really low where you'll have a region that's purified, and then it'll kind of come back to neutral, and then you'll see some noise of like some positive areas. But changing the windows, like you can't have a window size of 1, right? Because you need to calculate Ks, you really do need a little, you obviously need at least a codon's worth. And so changing the window size, there will be regions that, let's say, of your gene of interest that are like, no question, these regions are purifying selection, and maybe a few regions that are like, no question, neutral. But if you change the window size, you will get slightly different answers in other regions. Like there's more noise, and the noise is down at a scale where it's harder to feel as confident, if that makes sense.
Yeah, that helps. Thanks.
And Paul, if you have a better answer for any of my any of their questions, I'm happy to let you chime in as well.
Thanks. No, I think that was great. I might just mention too that, you know, when we think about kind of the impact of selection on a particular sequence, especially a coding sequence, you know, that strength of selection often varies over time, right? And so you can have regions, you know, that have been under like really strong positive selection, say 100,000 years ago, and you can still pick up some of that signal, but it's difficult sometimes to like deconvolute that from, you know, episodes of really recent selection, potentially at that same locus. And yeah, some other derivations of those, but no, it's such a cool and like intuitive concept and so powerful. But no, that was marvelous.
Any other questions you guys have? Okay, well, I will see you on Wednesday, and feel free to email me if you have any other questions.